Humans are testing the limit of most species survival with their planet-wide change, as urbanization, deforestation, loss of wildlife, and human-wildlife conflicts continue to spiral up. There's need to use every tool available to help protect what is left of the natural world. Molecular ecology is one such tool for conservation and can help in wildlife disease management and forensic in illegal trade. So what exactly is molecular ecology? As we will see, it is an interdisciplinary approach to some of the most fundamental questions in organismal biology. But some scientists today do not consider it to be its own discipline, but rather an approach taken in certain cases to answer certain questions. Most scientists, however, agree that it is distinct from other studies of organismal biology. Molecular ecology also defined as the application of molecular technique to answer ecological questions, and it is used to estimate population genetic diversity, to aid with wildlife breeding and conservation efforts, define species for conservation policy, track disease, and combat poaching. In modern molecular ecology, genetic data is analyzed in the context of field and observational studies to address ecological problems.
According to McCracken and Bradbury 1977, in the past, paternity testing was conducted through exclusion principle and possible to rule out the individuals as potential fathers if the exosome profile was not consistent with allelic variants in the mother and offspring. DNA fingerprint for plants is one of the applications for conservation and utilization of bioresources. TERI, a company that expertise in field of molecular markers, has worked the DNA markers on different kinds of plants. Through this project, TERI has collected different species of accessions and were analyzed using DNA fingerprinting techniques like AFLP and microsatellite. For this project, analysis of genetic diversity and identification of core accession for ex situ conservation and utilization can be achieved. DNA fingerprints are accurate as they spread out from nucleotide sequence differences between individuals. Plant DNA fingerprinting is complicated but detailed since it deals with populations and often more than one species. Because of the differences in the nucleotide sequence between individuals and large size of the genome, genomic fragments are generated and cultivars are discriminated by the differences in number, size, and sequence of specific DNA fragments. The genomic fragments are then employed as markers. Non-gene sequences are used as unique sequence markers. The picture shown above is the process of DNA fingerprinting. A molecular marker is a DNA sequence with a non-location on the chromosome and inheritance monitoring. Hoshiono et al. Stated that the evaluation of molecular markers depends on polymorphism found in the DNA. It works by highlighting differences of polymorphism within a nucleic sequence between different individuals. The differences include insertions, deletions, translocations, duplication, and also point mutations. The information obtained from DNA can be used to measure the relationship between organism and other genetic diversity.
Next, for molecular technique for conserving biodiversity is NGS with metabarcoding. It is a combination of NGS and universal primers for common barcoding region, used to assess conservation effort, land use planning, and assessing the functionality of ecosystem. So the PCR amplification affect the species detection way before metabarcoding became a routine in conservation of biodiversity and monitoring tool. Next is RAPD. RAPD stands for Random Amplified Polymorphic DNA. It is the PCR-based technique to identify variation of species of flora and fauna. RAPD markers can detect high level of DNA polymorphism and produce genetic markers. It is a simple and easy to perform when there is no prior knowledge about the genetics of the organisms.
The summary of the biodiversity information that is currently being collected using molecular technique is included the amount of diversity present and understanding on how population diversity is distributed in space and over time, an understanding of genetic distance and evolution, the degree of population relatedness, whether individuals are genetically identical or distinct, and the amount of gene exchange among individual and population. This information is essential for biodiversity management. A broad range of molecular techniques can provide this type of information. However, despite recent advancement and our growing understanding of plant genome, no single technique currently provides all of the information listed earlier. Usually, a combination of techniques is required to cover all of the information requirement, which increase the cost of using a molecular technique. Another limitation is that molecular markers are typically used anonymously. This has raised issues about the functional significance of the diversity shown by the marker technique. As a result, although molecular techniques have greatly improved our ability to characterize biodiversity, determining the importance or value of the diversity exposed and using the data in decision-making process remain difficult. The main reason for this is a gap between our genetic and phenotypic understanding of diversity.